Welcome back to the last video in the series for Unit 8, Aquatic and Terrestrial Pollution. This is Mrs. Gabriel presenting our 8.13 to 8.15 topics. We left off at 8.12 with an introduction to LD50 curves, so we're going to talk more about those response curves here. Okay, so I told you this last time too. This is a dose response curve. So the dose is how much an organism is getting, usually per pound of body weight or per kilogram of body weight. And the percent mortality is how many deaths there are out of, the total number of organisms that are being tested. We look at this with acute studies and with chronic studies. And an LD50 is just that place in the middle there where 50% of the total population that was tested dies at a specific dose. Okay, so I want you to look at the chart while I give you information that's also in the slides. I wrote it all out there for you. So we're going to practice by looking at the chart and envision everything on how we come up with these regulations for chemicals. So the EPA, Environmental Protection Agency, is who gives authority to regulate chemicals under the Toxic Substances Control Act of 1976. There are 4 groups of animals that are tested under this act. We can test animals, mammals, fish, and invertebrates. The logic is that if we know the sensitivity of each group, then we can protect them. The EPA uses results from these studies to determine concentrations that should cause no harm, are considered safe, using LD50 values, and then regulating allowed concentrations. So looking at the graph, using lab rats, you feed them a diet that contains different amounts of a pesticide ranging from 0 to 4. After feeding them these diets for 4 days, we count how many rats are still alive. We then calculate the mortality, which is the opposite. To find the LD50, we draw a horizontal line to the point of 50%. So here's where 50% of them died. Okay, so we draw that line for the 50% mortality. There, this line intersects. We draw a straight line down. It intersects the curve. We draw a straight line down to the x-axis, and we find that 2 milligrams per kilogram of body mass in a rat is the lethal dose for 50% of them. Remember that in order to determine the amount that is safe for mammals, well, actually, you might not know this. It's in your book. We divide this value by 10. So 2 milligrams divided by 10 is 0.2 milligrams. And then they divide it by 10 again to find an effective dose that a rat would be sensitive to. So the extra caution, we divide by 10 again to conduct a clinical trial on a human. So we really just divide the original LD50 by 1,000 before we look for a therapeutic dose in something for a human. So a dose response, remember, is not always looking at the lethal dose of 50% of the organisms. Sometimes we can just look at what is a sublethal effect, which means it's not lethal. It could change the organism's behavior or physiology or ability to reproduce. And we might use something called the ED50, the effective dose that causes the organism to display some kind of harmful but non-lethal effect. And we can also look at therapeutic doses, like with Tylenol or Motrin. We've done this, and those are dose responses that might be beneficial, like what is the dose where 50% would be effective or therapeutic, a T dose, where 80% of the people that took this got rid of their headache. So remember, in the U.S., the EPA authorizes and regulates many chemicals under the Toxic Substances Control Act. So your dose response curve is here, and I've explained to you LD50. We can also look at an LC50, which is lethal concentration, which is something we're actually modeling in a lab in class. We can look at that therapeutic effect, and there's this thing called the threshold dose. So the threshold dose is where we actually start the curve. As soon as we start the curve, where the dose can first detect an effect, that is what we call the threshold dose. And here are some screenshots that I took from AP Classroom for you that just reviewed some of the information I gave you. So just some of the big takeaways are that our dose response curve typically has a shape like what I'm drawing on right now, kind of like an S, right? And then remember where that threshold dose is. Remember that we measure in units of milligrams of something per kilogram of body weight, or something similar to a dose per gram or kilogram or pound of body weight for an organism. This is actually a graph that was on an FRQ years and years and years ago with copper sulfate concentration and brine shrimp. So they were looking at mortality of brine shrimp, and you had to describe things that were happening on this curve and even make some calculations. And here we have a researcher in a lab actually conducting a toxicology study. So again, the EPA is who's going to regulate toxic substances under the Toxic Substance Control Act, and they're regulating that with chemicals in food and cosmetics. Now we look at LD50s a lot, but we just look at lethal doses, doesn't have to be at 50, when we talk a lot about bioaccumulation and biomagnification. So remember, that's like the movement of the chemical in the environment going through, from one animal to another when it's eating in its food chain. And then the solubility is how well it dissolves in a liquid. So remember that we need to know more about the chemicals and what they're doing before they get into the organism. So we need to know a little about how that chemical gets into the environment before we even look at it in the organism. So how it moves into the environment that starts in the food chain. So solubility, which is how well a chemical dissolves in a liquid, and we know that that has to do with runoff, groundwater, right? Those chemicals getting in there and being grabbed by something that we're going to eat. And then fat soluble is not very soluble in water, but it does bind to soils. And those soils also underlie bodies of water. So bioaccumulation, remember, is that increased concentration in one organism over time. Biomagnification is that increase in concentration moving up the food chain. And persistence is a new word for us. That's the length of time a chemical remains in the environment. So remember, biomagnification is the concentration moving up the food chain. You can pause and read that on your own if you need a review. So Rachel Carson, remember, was the classic, she was the one that came up with the classic example of eagles and DDT and biomagnification. So DDT is not soluble in water. It accumulates in the bodies of organisms, and it moves through that food chain through biomagnification. Remember, with the top predators, they produce the thin-shelled eggs, and then the parents couldn't incubate the eggs. So DDT, we know, was what caused it, because then toxicology studies were made on this pesticide for us to learn more about its toxic effects on our sublethal doses on organisms. The concentration, remember, will increase if it's not going to break down, and that has to do with solubility. So as a chemical moves through the food chain, solubility matters, as well as in its environment. So some chemicals are going to dissolve really well in water, in the body even, and some are going to dissolve even better in oils and fats. So fish store mercury in fatty tissue, remember that? Okay, this is just a review again of bioaccumulation versus biomagnification. Look what I underlined. Biomagnification moves up that food chain. And here we're looking at the little red dots are supposed to represent like parts per million of a chemical. And then as you move up the food chain, the concentration increases. And here are the foods that you should avoid, especially if you're pregnant, because biomagnification is a real thing, and high concentrations of toxic metals are found in these specific fish. And then there are other fish, look at the list in the middle, that maybe you eat, or seafood, it's not all fish, right? We have lobster there. Maybe you eat this stuff, and these also have pollutants that bioaccumulate and then biomagnify through their food chain. So we can talk about heavy metals with these pollutants like mercury and other contaminants like PCBs. The persistence of a chemical refers to how long the chemical remains in the environment. Persistence depends on a number of factors including temperature, pH, whether it's water, soil, or even sunlight. We usually report persistence in half-lives. For example, DDT has a half-life of 30 years. So if DDT was sprayed on your lawn 30 years ago, half of it would be chemically degraded by now. But how much is left 30 years later? Still half of that original material. PCBs, think landfill trash. Okay, here's PCBs on the chart. PCBs take 8 to 15 years for a half-life. So 15 plus 15 is 2 half-lives, 30 years, but that doesn't mean that all the PCBs are gone. So that's a little bit about persistence. So if you look at the chart a little more up close, DDT we know is an insecticide, and its half-life is 30 years in the soil. So only half of it's gone, right? Half-life is 30 years in the soil. Roundup is a common herbicide. That means it like kills weeds, right? And Roundup has a half-life of anywhere from 7 to 70 days in water. Okay, so that's not as bad as 30 years for half of the life. Half of the life. And then we have vinyl chloride. We talked about that a lot in the last slideshow. And that lasts about 4.5 days in the air. So half-lives matter, and persistence of how long it lasts in the environment matters. Because these are all things, these are chemicals that are harmful to the environment and to living things. Okay, so if we know that something is hazardous at certain doses, and we know it can be harmful to us or to other living things, and it's really hard to get rid of it in our environment, then we have to consider a risk assessment, and then risk acceptance, and then risk management. So there's steps in this. So the risk assessment has to do with that dose response study. That's determining how much of the chemical we can handle being exposed to. And then we have to determine, like in your water quality report that you looked at, well, how much can we accept? How much are we willing to take a risk of being exposed to this? And then risk management, which is the policy or the legislation that we're going to put into place for what we will accept. And an example of that is arsenic in your drinking water. So we know that arsenic's in there. We know that arsenic has effects on human health. And we know that it's in a lot of the food that we eat also, and you can just tell by looking at this. But we accept a certain amount, and there is legislation that makes sure that it doesn't go over that amount. That's risk management. Okay, this topic is 8.14, pollution in human health. We're just going to cover a few here, but we want to identify some sources of human health issues that are linked to pollution. So pollution, these are linked to pollution. Dysentery, mesothelioma, and tropospheric ozone. These are linked to pollution and pollutant. Humans can be exposed to chemicals, pollutants, viruses, bacteria, things that are harmful to their health through their food, water, air, and anything in the building materials or household items where they live. And it can be difficult to establish a cause and effect between any pollutants and your human health. It's hard sometimes for us to figure out where it's coming from. We can't claim a chemical is harmful until we know how it behaves in the environment and how it behaves at different concentrations. Think of our LD50, LC50 lab. Different concentrations will tell us more about how the chemical behaves. This figure shows those roots again, the air, the food, the water, and add to it soil babies, fetuses can get exposed to through the mom, and which an individual might come into contact with a chemical, specifically bisphenol A. Bisphenol A is used in the manufacturing of hard plastic items like food containers and baby bottles, and it may be responsible for early puberty and increased rates of cancer. So you should make sure that you get bottles that say that they're BPA free, or any water bottles, BPA free should be on it. We know the exposure route, like the bottles, the toys, the food containers. So the next step then is to determine its solubility and its potential for bioaccumulation and biomagnification. Okay, so we don't have a point source here, but non-point sources such as untreated sewage coming somewhere but ends up in your streams and rivers, that's a non-point source, can make humans very sick. It can cause dysentery, and dysentery is inflammation of intestines, and it's caused by a bacteria. So dysentery, bacteria, dysentery, bacteria, dysentery, bacteria. It will cause bloody diarrhea and abdominal cramping. And it comes from contamination in untreated sewage, which usually ends up in streams and rivers. Dysentery is not something that's predominant in the United States because we treat most of our sewage before it's released into any kind of stream or river. However, in developing countries where those water treatment facilities and sewage treatment facilities don't exist yet, dysentery is very common. Mesothelioma is another human health concern that has to do with pollutants, and this one in particular has to do with asbestos. And you can see asbestos close up. These are the fibers from asbestos. So it's a rock, and then we pulverize the rock, and we've used it in insulation materials in buildings, and we've used it in tile, so fire and flame do not go through it. But these little fibers, when inhaled, if the fibers are inhaled, can eventually lead to cancer, and the type of cancer is asbestos. And you can see how the asbestos fibers in the pictures lead to different types of mesothelioma, and it also can inflame and damage cells of other tissue in the heart and in the abdomen. Respiratory problems and overall lung function can be impacted by elevated levels of tropospheric ozone, which, if you recall, is a secondary pollutant. So tropospheric ozone is the ground level ozone, and it's formed from volatile compounds, nitrogen oxides, and sunlight. It's a photochemical reaction. So the EPA developed an air quality index, which is right over here, air quality index, that we use to notify people when the tropospheric ozone levels are harmful and going to create respiratory distress. So make sure that you remember the AQI. AQI is the air quality index that the EPA has developed in order to create an awareness for tropospheric ozone levels that are going to aggravate and instigate respiratory health. And here's a close-up look in the upper right corner. So ozone in the troposphere can constrict muscles in our airways, and this is what it looks like. So right over here, my pointer's looking at the side view of a healthy lung. And then, well, we're actually looking at lung tissue, a bronchial tube, and then these are your alveoli. So when the lung gets inflamed because of ozone, when it gets inflamed, it's harder and harder to bring air in. And then any particulate matter that might be in that air now has less room and space to get through, and it's constricting our airways. And then you add particulate matter, which is usually commonly found when we have high ozone levels, and that's just a recipe for disaster for anyone with lung problems and respiratory problems. And then you have a close-up view of what that looks like as the bronchioles start getting smaller and smaller and smaller. So the little tubes that the air passes through is getting smaller and smaller, more narrow, and breathing becomes more and more difficult. Okay, so here's a practice question that was on AP Classroom and some ideas to take away. Make sure you pause it and practice it yourself. Last topic for Unit 8, 815, pathogens and infectious disease. Okay, so you have to be able to explain human pathogens and their cycling through the environment. So pathogens are plague, tuberculosis, malaria, West Nile virus, SARS, MERS, Zika, cholera. These are some of the big ones that they expect you to know about. So these pathogens can adapt to take advantage of new opportunities in order to infect and spread through human populations. And we learn about them in environmental science because these are often related to climate change and changes in climate zones because of global warming, and they're also related to sanitary conditions. So there's both environmental topics, and then we're just making the last bit of connection to its effect on human health. Okay, take a look at the map and just notice where the yellow is. Yellow is our current distribution of one type of malaria. So we'll learn more about malaria in a few minutes. And scientists are predicting with climate change that what you see in red is going to be suitable climate for vector and parasites by 2050. So it's spreading, and notice where it's spreading. So the climate zones spread north and south. So the tropical zones and the subtropical zones are moving into our temperate zones. And with that, pathogens, infectious disease, and all the vectors associated with them are also spreading into those zones. And poverty-stricken, low-income areas often lack the sanitary waste disposal and the technology in order to protect themselves from the increase in these pathogens moving into their zones. So what are our pathogens? Okay, look right where my laser pointer is. Examples of the most common pathogens that we are referring to are viruses, bacteria, protozoans, worms, and fungi. Sometimes we call them infectious agents. Sometimes we just call them germs. But pathogens, we know, adapt. So as the climate zones are moving more north and more south, those pathogens are also moving with them. And we've noticed by collecting data, and this graph, by the way, is by the CDC, we've noticed that disease cases from infected mosquitoes, ticks, and fleas have tripled in 13 years, tripled. So pathogens can occur in many environments regardless of the appearance of sanitary conditions. It doesn't matter how clean you are. A lot of these things can still spread through our environment. This is why you should care personally about you and washing your hands. Okay, malaria is the first disease we're going to talk about. Malaria is a parasitic disease. It's a parasite, guys, and it's mostly in sub-Saharan Africa, mostly found there. But we're going to look at this map in the lower left corner. So the map on the United States shows how the mosquito as a vector for the pathogen of malaria, the parasite that causes malaria, the disease, has been spreading more and more northbound. And that is because the subtropical areas are expanding because of climate shifts. Now, the mosquito that we're talking about, look at the picture in the upper left corner, is actually a carrier, right? So if you look at the picture to the right corner and you look at the first person in red, so that person has the disease. So an uninfected mosquito can bite that person, and when the mosquito bites that person, if that person is infected with that pathogen, the mosquito can pick it up. And this is not with all diseases, but this is why it's important we understand how vectors work. So then the mosquito now has the infected parasite and can transmit it to any other organism that it bites. And that is why the mosquito is a vector. And remember, malaria is a parasitic disease. It's caused by bites from infected mosquitoes only, and it's most often found in sub-Saharan Africa. So if you look at the map in the top, and I know some of it's covered, and just find Africa, you'll see that Africa is a lot of sub-Saharan, and more of Africa is red for high risk for malaria distribution, as I just told you. But look where else we can find malaria being carried by mosquitoes as a vector. So remember, as the climate is shifting and we're moving further and further north and south with our subtropical zones, these mosquitoes that do really well in the subtropical climate are also going to move with them. And then I thought it would just be fun for you to look at the different diseases by mosquitoes. So if you look at the chart that's over on the right, I showed you a picture of two different mosquitoes. One is the type of mosquito that carries malaria, and the one on the right is the one that carries what you're probably most common with, which is dengue or yellow fevers. So the interesting thing about these charts, these mosquitoes have different types of niches. So if you look at when do they bite, the mosquito that transmits malaria, that's a vector for malaria, bites more at night, but the mosquito that transmits the parasite for dengue and yellow fever bites more during the day. And then look at their breeding ground. So also different niches. One is in any body of water, and the other one is at like a shallow water surface. So it could even be just a puddle. So malaria is a parasitic disease. It's caused by bites from mosquitoes. West Nile is also transmitted to humans by infected mosquitoes. However, West Nile virus is a virus. West Nile is a virus, not a parasite. So the mosquito is still the vector, but in this case, the transmission is a little different. So this actually can move from animal to mosquito to human. And if you look at the picture, we can follow that vector. So the mosquito again is the vector, okay? And then we'll just follow, if you look at the mosquito first. So an insect vector, and the mosquito bites the bird, and then the bird actually has the infection now, and the mosquito can then transmit it to the host, which is people or other mammals even, So the West Nile virus is then transmitted back and forth between the birds and the humans. So the birds can host this organism, and then it can spread to humans, but it spreads through the bite of a mosquito. And then if you look at the map of the United States for 2018, this is the incidence per 100,000 people. So if you look at what's black, look at the black states, you'll see where West Nile virus is more likely to be transmitted. So in the darkest colors. And the report is in per 100,000 people. And then look for Maryland. We're pretty dark, so we're also at high risk. So their niches are specific to definitely central United States, north and south. So why do we care about malaria and West Nile virus? Well, if you start having these symptoms as the climate is shifting and getting warmer, you might want to consider getting tested for having the parasite that causes the disease of malaria or the virus, West Nile virus. So the symptoms are listed here, and I'm not going to read all of them, but I will tell you a little bit about malaria. And both of them, the symptoms show up within, within a week for malaria and up to 2 weeks for West Nile virus. And you can get tested for them. You would take an anti-parasitic and bacterial biotic and an antibiotic for malaria. But West Nile virus, you kind of have to ride it out and make sure that you're treating yourself good for dehydration. But notice that you can have things that feel like flu symptoms with them, and you should get them treated. Okay, the plague is something that still does exist. A lot of people don't talk about that, but we do have a couple cases in our country every single year. So I just thought it would be fun to look at the old-fashioned plague doctor. Think about why he's dressed that way while we explain a little about what's going on with this disease. So this disease is not parasitic and it's not a virus, it's a bacteria. So a bacteria, and the vector is a flea. So the flea carries the bacteria, and then the rat carries the fleas in this image. It doesn't have to be a rat though. It can also be other small mammals that are usually densely populated and typically found in a little bit more rural suburban areas. So traditionally they were found in ports, like ports where ships would come in, because the ships had a whole lot of rats on them, and that's how the disease just started spreading. So the disease was more predominant in port areas historically. So the rat carries, in this scenario, the rat carries the plague bacteria. The flea bites the rat and picks up the bacteria, and then the bacteria multiply in the flea. They do real well in the flea as a host, and then when the flea bites the human gets infected. And I'm going to show you in a minute what that looks like if you get the plague. So it's pretty disgusting and scary, like you can get black splotches on your skin. So imagine if you're a doctor and you're in a port town where you have to treat people that have symptoms of plague. Well, they dress this way. It's kind of like the old-fashioned coronavirus masks, right? They were covering their face. It's just, they look so creepy and scary. So the flea is the vector that carries this pathogen. The flea is the one that bites you, but the plague is caused by a bacteria. So I also showed you 3 clinical forms of plague in the pictures below. And one is bubonic plague, you've probably heard of it, and then septicemic plague and pneumonic, like lungs, pneumonic plague. Plague symptoms depend on how the patient was exposed to the bacteria. And then you should look at this map. This is right from the CDC, Centers for Disease Control website, of where plague has been reported in the United States from 1970 to 2018. And notice where the cluster is, like that New Mexico side and California side of the United States. So imagine if you had all this black formation on your skin and the doctor sees you, how they felt when they walked up to someone that had this, knowing what the symptoms are going to be. So people do die from all these diseases. Remember, plague is formed from a flea bite. Now, just like plague, tuberculosis is also a bacterial infection. You may have heard it called TB. Tuberculosis, you can actually get treated for if you get it and you catch it early enough. So this is highly, highly contagious. Droplets from coughing and sneezing from an infected person spread, and easily you will get it. Now, if you look at the map, you can see, and again, they're reporting this in per 100,000 people. So these numbers are just per 100,000, not total population. So you can see, again, where we're going to see more tuberculosis. So it's usually where we don't have as much health care, and it's usually in more poverty-stricken areas. But don't be fooled by that, because, and this is why we learn about this stuff, just like plague, just like malaria, you can still see that some people somehow get contaminated with these pathogens. So tuberculosis attacks the lungs, and you'll cough up blood. That's one of the signs of tuberculosis, coughing up blood. And remember, it's a bacterial infection. Ooh, and here's another bacterial infection that can be transmitted in our environment, cholera. Now, the symptoms of cholera are that you'll have very watery diarrhea and vomiting, and you become severely dehydrated because of the watery diarrhea. And then severe dehydration also leads to a low blood pressure and rapid heart rate. So when people die of cholera, they're really dying of severe dehydration. The numbers are ridiculous with the number of people in poverty-stricken areas that get cholera infections and die. So cholera is a bacterial disease, and it's mostly found in, the bacteria is mostly found in any water that might have sewage in it. So you are looking at people washing their dishes in shared water, and these people are going into the water, so their bodies are going in there, and their bodies might have some of the same material as what you would find in sewage, and they might be going to the bathroom in the water, or someone might be going to the bathroom on the water's edge, but that is going into the water and they're washing their dishes with it. So the bacteria is then in the water, so we're calling it infected water. So you can get it from dishes, you can get it from foods, you can get it just from drinking the water and not completely sterilizing the water. And it's a bacteria, and it's called cholera. And it's definitely related to sanitary waste disposal. So we just talked about a whole bunch of pathogens. And remember, the big takeaway is pathogens can adapt to new conditions, and they can spread through the environment with climate change. And remember that pathogens can occur in any type of environment. It doesn't have to be sanitary or unsanitary. Really, pathogens can be found anywhere. And they spread Look right in this here. They spread in many different ways through what we eat, through what we touch, through what we breathe. Pathogens. Okay, I don't know if you've heard too much about these SARS and MERS, but they're coronaviruses. They're another type of virus that are spread very easily, can make us very sick, and just like all the other diseases here, can kill. They can lead to death. So these are two other types of respiratory infections, and they are airborne. They are transmitted through something in aerosols. So someone coughs, and as you can see from the picture down here, their little droplets are breathed in by another person, and voila, the disease is transmitted. SARS and MERS. So they're viral. They are viral. And these can be transmitted from animals to humans. Animals to humans. Now, when a disease originates in animals and spreads to humans, it's called a spillover event. So as I said, SARS and MERS are both types of coronaviruses, and COVID-19, they originate in some other animal. SARS and MERS originated in bats, and they spread to animals that were closer to people, like camels, and then can spread to humans. So they're coronaviruses, they originate in other animals, and they spread to humans. You can pause the screen and look over all the essential knowledge statements that I just discussed in more detail with you, but we are done. So welcome to the end of Unit 8, the last and final video. We just finished up to 8.15. If you have any questions, feel free to stop by and ask me whatever you need. This is Mrs. Gabriel signing out.